before. I actually tried to do this kind of research back in April because I suspected there would be serious overlap between some of the target sequences they're looking for with the PCR test and our human genome because of this confusion about exosomes and what RNA comes from what source and all of this. I looked at a different protocol than the one published here. I looked at one from Germany, and this one is from the Pasteur Institute in France. But it really confirmed my suspicion, and I was glad to see it, that basically one of the primer sequences in the PCR test, according to the Pasteur Institute protocol, is an exact match for a sequence in our own human DNA on chromosome 8.
There's not enough viral RNA to detect directly in the patient's sample, so a process called reverse transcription polymerase chain reaction, RT-PCR, amplifies many copies of a segment of the N gene.
So what they're saying is that there's a tiny, tiny amount of the RNA that we're looking for in this sample that's really messy and contains a lot of stuff. So in order to be able to detect it at such tiny, tiny amounts, they have to amplify it or make multiple copies. And that's really what the PCR test does.
Short, single-stranded pieces of DNA called primers recognize unique RNA sequences within the viral genome that bracket the target region of the N gene. After the first primer binds, an enzyme called reverse transcriptase extends, synthesizes, a single-stranded DNA copy of the viral RNA.
Okay, so I want to stop here because this really shows the important part that we need to pay attention to. That you see in the bottom right hand, there's this short primer, right? So that's the only thing that we have identified in advance, like we created the primer. We could make a primer for any sequence, for any organism. And then when we put that in this reaction, basically we're looking for a piece of genetic material from some organism that matches it, is the complement. Okay, because when these two strands go together, they have a complementarity. It's like a plus and a minus that attract each other. And it's G's and C's and A's and T's. Okay, so this is going to look for a specific sequence in the sample that we're trying to find. And what the long strand that you saw in this video is, that's the sequence that could be from another organism that we're identifying. Now, since the primer, at least one of the primers that we have been talking about, is an exact match for a sequence in chromosome 8, then it could identify a piece of a transcript from chromosome 8 rather than some genetic material from another organism. In other words, it can detect our own DNA. So I'll just want to share one more thing here to verify to everyone, because it's really important to make sure that this information is correct. And so this is right here, the sequence of interest, okay? This 18-letter code, and it says it's from the RDRP gene in the virus, okay? Now, I don't know if I can show both of these on the screen at the same time, but I'm going to try my best. So this I'm scrolling through right now is from GenBank, which is part of the National Institutes of Health, where they store all of the sequences from the Human Genome Project and also from other animal genomes. And so you can see Homo sapiens is human, chromosome 8, and then all this gobbledygook here is just basically how they took all the DNA and split it up for sequencing, and this is the primary assembly when they did it this way. But if you look to the bottom of this, and it gives some references, here's the actual sequence, and that matches exactly this sequence here, which is from, sorry, you're in the way, the, where does it say? The Pasteur Institute, sorry, it's listed on the top in the file name. But this is the Pasteur Institute protocol from the World Health Organization. So you could see those sequences match up perfectly because there's more than one primer that they can use. So So this whole testing thing is very, very heterogeneous around the world. So in other words, there are different protocols in virtually every country. And a lot of them are recommended through the World Health Organization, but there are some from other bodies too. And they can use different primers. So, and even when they do just one sample, they're not going to use just one primer for a test and say if that one's positive, it's positive. So it's not exactly that clear cut. But what we're talking about here is at least one of the primers that they're using in one of the tests for our own sequence. Okay, so this could come up positive in any human regardless of the presence of any virus or anything else. So it adds another level of confusion and obfuscation to how do you interpret the results of a test like this. Now, I still think the most salient point is that we don't know where these sequences came from because there was never a purified particle of a virus where they had a bunch of them together in one sample and they could characterize those and pull the RNA material right out of those particles. They never did it that way. They only looked at it in this dirty sample, which was a mixture of many, many microorganisms and human cells. And they pulled these sequences out of there because they thought the sequences belonged to the family of coronaviruses. But that was based on only about 80% sequence identity between those samples. And, you know, when I looked and did that research previously with the other probes for a different protocol, for the Germany protocol, I found that many of those primers in that sequence had 80 to 85% sequence identity with stretches in the human genome as well. I just didn't find a complete match. I did find 18 in a row that matched, but those primers were 24 bases long. So there's a lot of overlap in this. And then if you also look at the nature of the PCR test itself, is that the enzymes that they use to copy the RNA make mistakes. And so they could give you a lot of false positives. And this is one of the problems that's always been known about this test. And the more you amplify it, because you do this test in cycles, right? And each cycle you double the copies of that piece of RNA that you're looking for. And the more times you double it, the more false signals that you get, because you're also amplifying like the noise signal, which could be other sequences that are partially matched, and they're not detectable until you amplify them so many times, and then it's like everything that is an artifact in there is also detectable, and that gives you a false positive. And that's one of the main reasons why Carrie Mullis did not recommend using this as a diagnostic test, because of all those false positives. But, you know, in my opinion, since we don't know what the origin of this sequences that they're testing is, other than now we know one of them may come from our own sequences, like the human sequence, then there's really no way to calculate any accuracy. So I would say, you know, there's 100% error rate with this test.